Hey biology classes, welcome to our first online lecture. This is going to be a little bit weird, so you guys will just have to tell me what works and what doesn't work. I'll post these videos and I'm going to put them in short increments so that you guys can watch them kind of at your own pace. If something is not helpful or if something is super helpful, if you guys will tell me in the comments on PowerSchool, it'll help me make the videos more useful for you guys. So make sure that you guys use these with the slides. You're going to want to take notes probably from the slides that I posted online and then use this to supplement those notes. I'm going to do all the sketches that I would normally do in class on these videos. All right, so let's get started. So as far as environmental influences goes, there's a good picture of this in the slide. This is with Siamese cats. They have the dark coloring on their faces, and that turns out it's from temperature. So scientists actually came up with this crazy idea to simulate this experiment by shaving a cat. They shave the cat and then they put ice actually on the cat's skin, and as you'd expect, you can, if temperature really does cause the hair to grow in darker, when they shave the cat and put the ice pack on, the hair should come out darker. That's exactly what they found happen with Siamese cats. So some things are genetic, some things are environmentally influenced. Same thing with human height. You are genetically predisposed to be a certain height, but depending on nutrition and things like that, your height can also be affected by that. So keep in mind, just it's not 100% genetic. Just to get rid of one idea out of the gate, there's no such thing as blending inheritance. We like to believe that this is the case because we see it every day. If you take someone with really dark hair and they have a child with someone that has blonde hair, you might get an in-between color of hair. Those are mixed traits that come from multiple genes. Those are not genes that blend, okay? So just kind of try to get rid of that idea out of your head. Blending inheritance, not a thing. The first kind of genetics that we're going to start with is what we call Mendelian genetics. If you guys were at Ridgeview in 7th grade, or even if you were somewhere else, you've probably already learned about some of what Mendel had to say with genetics. If something follows the rules, basically, we're going to call that Mendelian genetics. We'll get into a little bit more complicated stuff later, but for today, we're just going to cover what Mendel discovered. Mendel's a monk. He looks at traits in pea plants as he's growing things in the garden. He's basically just identifying 7 different traits that he's going to look at. And the 7 traits that he looks at, I list here in pea plant traits on number 2. Those are, let's figure out how we're going to do this. All right, so those are seed shape and color, so they're green or yellow. Pod shape and color, also green or yellow, and then smooth or wrinkled pods. Plant height and flower color, so purple or white flowers. The really important thing about the traits that Mendel picked is that they come in exactly 2 traits, and then also pea plants can self-fertilize, which means you can cross a plant or mate a plant with itself to see what traits it has. We'll see how that's important a little bit later. Alleles, we've already covered this, but we are just going to go over it again. But alleles are the 2 different forms of a gene. So for example, tall or short, those would be 2 different forms. Purple or white would be 2 different forms of a gene. Those would be called alleles. Just a review of chromosomes there at the bottom. Eukaryotic versus prokaryotic chromosomes. All you need to know is that bacteria have 1 looped chromosome, and eukaryotes tend to have numerous chromosomes of different levels. We have 46. Some things have even more chromosomes than that. And then other things might have just, for example, chimpanzees, they would have 48 chromosomes. So the number can vary, but just know that eukaryotes have more than 1, whereas prokaryotes are going to have 1, and then that they look different. Karyotypes. I'm not going to go over those too much because I'm going to have you guys do an activity online for those karyotypes, and it'll actually walk you through what a karyotype is and what it looks like. So we're going to skip that for now, just for the sake of time, so you're not doing things more than once. We're going to talk now about probability. So one of the things that Mendel really did is he enabled us to be able to use math to figure out how genes were going to turn out. So he saw that traits are inherited in predictable patterns, and he allowed us to make predictions. This is where we're going to come to everybody's favorite thing, Punnett squares. So first, we have a few things we need to know before we can get to actually doing Punnett squares. And that the first thing is just going to be some genetic terminology. So P, F1, and F2. Let's just go over here and we're going to map out a couple of generations. So first we would have our parent generation. These are going to be the two plants that we would mate together to start with. They don't necessarily have to be plants. We talk about this with animals also, but I'm going to show you plants because that's what Mendel used. Parent, we would put a capital P there to stand for the parent generation. For parent generations, we're always going to use things that we call true breeding. So true breeding means they have the same alleles. So let's say that a plant had alleles for tall or it had alleles for short. We cannot use heterozygous things. So we would not use a big T, little t organism here, one that had one of each trait, a tall allele and a short allele. We'd want to use one that has the same two alleles. So for our parent generation, let's just say for this parent generation, I'm going to do exactly what I wrote over here. I'm going to use one parent that has all tall traits, so big T, and I'm going to cross it or mate it with another parent that has both short genes. So a tall pea plant with a short pea plant. Two tall alleles, two short alleles. So each parent has only one option for the allele that it can donate. Now each parent only donates one allele. So this parent can either give this big T or this big T, and this parent can either give this little t or this little t. That's going to create what we call a first filial generation. So filial, if you've been paying attention in Latin, this means sister, brother, depending on the ending that you put on it. Sorry, that's wrong. Ignore. Daughter or son. That's better. So this means daughter or son, depending on the ending that you put on it. So these are going to be our daughter plants, or daughter generation is usually what we call it. But basically, if we mate the parents, then we get daughters. So first filial is going to be abbreviated F1. And so we just take filial 1, that's the first generation. So we cross these together, we get our first generation. If you look at this, you can tell what they should come out as. If this can only donate a big T, then our first filial generation has to have one big T. And then this guy can only donate little t, so they have to have a little t. If we mate this now to itself, so we're going to do this, and I'm going to put an S over it just to indicate that it self-fertilizes. So we would take the pollen from one of these flowers and put it on the pistil of another flower. And so we would basically pollinate this flower with its own pollen. So we only have genes from this flower. We're going to get these two genotypes then crossing. So we would get now a plant that has the option to donate either big T or little t here and big T or little t here. So now for our F2 generation, we're going to have some different options. And that's where the Punnett square is going to come in handy. And so we'll talk about that in just a minute. Before I get to Punnett squares, though, for determining what our F2 generation is going to look like, I want to go over what it means to be dominant or recessive and how we mark that in genetics. So Mendel got lucky when he chose the traits in that they only have two forms. They can either be, purple or white, tall or short, wrinkly or smooth. There's no in-between trait. So what that means is that one of the traits has to take over if we have a genotype like this where both alleles are present. So what we're going to do for this is dominant traits are going to be the one that can mask the other form. And so the dominant trait is the one that takes over. So for example here, tall is the dominant trait. If you have one allele for tall and one allele for short, the allele for tall will take over and you'll get a tall plant. So we would mark that like this. You guys actually should have this in your notes already. See if I can do this without knocking my video over. Okay, so you should have T would be for tall pea plants and little t would be for short pea plants. So the big letter is going, or the capital letter is going to be used for the dominant form and the lowercase letter is used for the recessive form. So that means if we had both of these present, like we do in this individual, that plant would be tall. And we're going to keep that consistent for everything. We're always going to use capital letters for dominant traits and we're always going to use lowercase letters for recessive traits. Let's talk genotype and phenotype here for a second. So genotype and phenotype, bear with me as I learn to use technology. All right, so genotype and phenotype are going to be a combination of alleles that has a trait for. So let's start over, sorry. Genotype is going to be a combination of letters, and phenotype is going to be how that organism actually looks. So the genotype is going to be expressed always as a written pair of letters. And the physical expression, so how that actually shows up in the plant, is going to be an appearance. So our genotype would be these letters, okay? Our phenotype is going to be tall. It's going to appear tall. So its genotype is going to be this combination. Its appearance is going to be this. So if I ask you for the phenotype in a question, you're going to want to tell me what the animal looks like. If I ask you for its genotype, you're going to want to give me the combination of letters that would make up its genotype. So big A, little a, big A, little a, those sorts of things. Make sense? Okay, one more thing I'm going to show you guys in this video, and then we'll cut off this one for a new video. So, homozygous and heterozygous genotypes. Homozygous is going to have the same two letters, so either big T, or little t. Both of these are the same, right? We have capital, or lowercase. That would be homozygous. So keep in mind, using prefixes can be helpful. Homo means the same. Hetero obviously means different. So heterozygous individuals would look like this. They have one of each type of letter, one capital, one lowercase. So if an organism has two different alleles, it's going to be heterozygous. If it has the same alleles, it's going to be homozygous. We're going to start with crosses on our next video, and then I'm going to give you guys some practice problems. I'll walk through a couple of the problems with you on a video to show you what to do, and then I'll assign those as homework in the homework block on PowerSchool. Let me know if you guys have any questions, and I will talk to you again soon. Bye.